Okay. So today, what we're going to do is move on to our second unit. The unit 1 test, of course, is Thursday. We're going to move on to talk about the material in unit 2, and we're going to start with growth. And I think growth is a really fun one. Hang in there. There's a lot to know, but it is very concrete and accessible, and you're going to do stuff in lab that really relates to it. So we're going to talk about how we grow microbes, and then how we understand the theory of microbial growth. So these are the 4 basic questions in this, like what do microbes actually need? What conditions do they need? And then how do we use that information to grow them in the lab, or conversely to kill them in the world? How can we measure growth? How can we determine if we were successful in growing them, or if they're growing where we don't want them? And then we'll talk about some sort of mathy things around this as well. The goal of all of this is in part, at least, to control that growth, either to prevent microbes from doing things we don't like hurting us or making our food go bad. Understand places in, like what they're doing in the environment that are important, or things that we might be using them for. And also, if you want to, for instance, use microbes to make beer or make cheese, you kind of need you need a lot of control over what's happening if you want to get the desired result. Now, with that said, we're going to focus largely in this section on bacteria. And what we'll talk about will also be applicable to yeast and somewhat to molds, but not really to protozoa or algae, because their sort of growth is different, in part because they're not prokaryotic, although yeast are eukaryotic and they still adhere to a lot of these rules. Now, you've already looked at what it looks like when you sample mixed culture from the environment. And so here we have in lab, we collected these on Petri dishes, right? And this particular Petri dish is about 10 centimeters across, which makes its surface area 1/15 of a square foot. There are little ones, little baby cute ones. You'll get a chance to do that for the transformation lab. And then there's giant honking ones. And what are these called? Write that into the chat. We're just kind of getting warm up here. What are these little growths? When we look at the plate and we see stuff growing on it, what do we call this? Circular growth. That's right. Yep, that's a colony. So how did it get there? Like, what formed this colony? Where did it come from? Yeah, that's right, Carlo. It was one cell, an invisible cell, that landed on here. Either you put it with a cotton swab, or it landed from the air, or came in water. But that one cell grew and grew and grew. It went through multiple cell divisions, and now there's a big mass of cells. I would like you to take a guess, okay? And there's no abstaining. You're not allowed to put a question mark for this one. How many cells are in that colony, do you think? Take a guess. I'm not going to remember what your number was. So just think about it. Put a number in the chat. Throw that wheel in the roulette table or whatever. I'm not a gambler, so I don't know the terminology. Great. Yeah. So roughly millions or hundreds of millions or billions are in here. Okay, this one little dot has a lot of cells in it. Now, how long did it take that one cell to produce millions of cells? How long did it take those cell divisions to occur? Yeah, it was a couple of days. And it, depending on the species, might take longer or shorter, but that's a good period of time for most of these species. As you can see, this was produced after about a 48-hour incubation period. I think the question is pretty obvious, right? Is this a pure culture? Nope, it is not. We can see that these cells, however they're growing, whatever they're doing, as a group, they have distinctive features. We can say it's very likely these are the same species, but these are definitely not. They're not the same color. These are not, they're not the same size. These are not, they're not the same texture, and so on. So there's multiple species, at least a dozen. We don't even know if these two colonies that look so similar are actually the same species. They might be just close relatives or not even related at all. From your prep, you will have learned what the categories are of conditions that we focus on. But what are those conditions that we care about when it comes to growing bacteria that we have to know about or pay attention to in order to really have some control over an understanding of their growth? Just throw any of them into the chat and I'll read them out. Yes, temperature is a big one. Oxygen level, pH, water, time, okay. Nutrients, yes. Osmotic pressure, exactly. Affection. Great, yeah. So lots of different things to pay attention to when you're trying to grow microbes, but temperature, pH, how much salt basically, or sugar or stuff that water likes is in the environment, the osmotic pressure, whether or not the atmosphere has oxygen or carbon dioxide or other things, and what nutrients are provided. Now, when you look at how organisms grow in different temperatures, there are categories. So these categories relate to what's kind of the favorite temperature and then sort of the upper and lower limits of where particular organisms can grow. And so organisms in these different categories have been given category names. So as I indicate one of these lines, you're going to write into the chat what you think the group is called. So this purple line, where the organisms grow best at like maybe, cold-ish temperatures. Some of them can survive in ice. Maybe they're sort of dragging out a living at room temperature, but they like it pretty cold. What do we call them? That's right. We call them psychrophiles, cold loving. Cold loving phile means love, psychro means cold. Now we've got these guys that prefer room temperature. They can kind of hack it, not at human body temperature, but almost. And they're barely alive in ice, but they really like, just sitting on your counter. Who are these guys? That's right, psychrotrophs. These are cold living. All right, now, of course, we've got the guys that like human body temperature the most. They might be able to make it a few degrees into the 40s, and they're dead below about 15. So we call these guys mesophiles, exactly. Now, how about these guys? They like a good 65-degree day, which would boil us. They can live, pretty hot. At human body temperature, they're going to be way dead. These are thermophiles, heat loving. And last but not least, these guys can live in boiling temperatures. Why is that? What do we call them? Indeed, these are the hyperthermophiles, extremely hot. Which group contains the most human pathogens, or pretty much almost all human pathogens? Hopefully it is obviously going to be the mesophiles. Now, food spoilage can be from a number of species in different categories, but interestingly, it's a lot of psychrotrophs. Okay, so, and this is just straight out of your prep, just to remind you of stuff you should know. You can go look that up and memorize that to your heart's content. Let's talk a bit about what's actually happening at the cellular and molecular level when an organism is affected by or responding to temperature. Why does temperature matter? What in the cell is actually affected by temperature? As temperatures get really hot, what happens? As temperatures cool down, what happens? What in the cell is being affected by temperature? Right. Yeah. Indeed. So I would say the two good answers, and maybe I'll actually write them in so you can kind of have them in the video. The two things that are really affected by temperature are proteins, and that includes a lot of enzymes. Okay, so biochemistry of the cell is going to be affected in membranes. Okay, so whether leaky or rigid. And then what about this? Why is the graph asymmetrical? When we look at it, we can see there's an optimal temperature, there's an upper temperature, and then a lower temperature. But as we deviate from this optimal temperature, high temperatures very quickly cause catastrophic failure of the organism. Low temperatures more gradually reduce viability. Why is that the case? What's happening in the too high heat situation that's different from what's happening at the low temperature situation? Go ahead and raise your hand if you have an answer to that question. No one wants to be brave. Any hints or thoughts, put them into the chat. What's happening at high temperature that's different than what's happening at low temperature? What does heat do that the cold does not do? It is a chemical change, Lucy, that's right. What does heat do to those proteins? Yes. Let's be more specific. Yes. Heat denatures proteins, including all enzymes, and causes membranes to leak. Whereas cold slows enzymatic reactions and causes rigid membranes. Now, these are actually different things, and as it happens, the denaturation process is more rapid. And so you get a much faster loss of function and also loss of life as heat goes up. As we slow things down, we cool them down, biochemistry is still working, just more slowly. So we're just getting a slower rate of growth. But there is a point after which the enzymes can't move at all, the structures can't move at all, and the membranes cannot exchange anything because they're too rigid. And at that point, the cell will die. If we think about extremophiles, and again, this is a very, thinking about extremophiles is sort of human-centric, right? We think of ourselves as being the gold standard for living things. So when we compare our mesophiles, which are similar to humans, mesophiles compared to thermophiles, how have thermophiles adapted or changed their nature in order to survive higher temperatures than we can? Use the letter or letters. It's a select all question. Put that into the chat. Again, you're not allowed to put a question mark for this one. Just choose some letters. Even if you're not sure, take a guess. It will help your brain to remember. Excellent. Keep those answers coming. Good job, everyone. Excellent. Okay, so let's talk about it really quick. We have a problem, which is as the heat goes up, the tendency is for proteins to denature. Their hydrogen bonds that hold them together fall apart. So genetically, thermophiles are different from mesophiles in that the genes code for more bonds in between the different amino acids of the protein to make them more rigid and withstand higher temperatures. They've also reorganized their membrane so that their phospholipid fatty acid tails are more saturated, more straight, so they pack together closer to resist the tendency of heat to drive them apart and make them too leaky. Okay. On the other hand, psychrophiles, how are they adapted differently than mesophiles? They live in the cold. Compared to a mesophile, how have they been altered? Put your letters into the chat. Yeah. The problem with those cold temperatures is that the proteins are now naturally more rigid. So we've got to compensate that by having genes that code for looser proteins. And the membranes tended to be rigid. So we are compensating for that by introducing, biochemically introducing space in between the phospholipids by bending those fatty acid tails, making them unsaturated. Okay. So when we think about temperature requirements, we know that it is effective to freeze foods to preserve it, but doesn't preserve it indefinitely. So let's think about why that might be. We can't keep things in the freezer forever. It's not actually safe to eat stuff that's been in the freezer for a very long time. Why is that the case? Why doesn't freezing food kill all the microbes? Why does the food eventually spoil? Okay, that's part of the answer, and that's a good answer, Spencer. The food could have endospores that could still be have viability once the food is thawed. So you still have to cook it properly, and you don't kill everything. If they've got endospores, they're not going to die. And then there's microbes that can actually still grow at that temperature. It's effective, though, at preserving food and reducing foodborne illness, quite effective, because what it's doing is slowing things down across the board, right? And the things that are likeliest to make you really sick are not going to survive at this temperature unless they have endospores. Okay. All right, so next question. Mycobacterium tuberculosis, causative agent of tuberculosis, and Mycobacterium leprae, the causative agent of leprosy, are interesting because they're very close relatives, but Mycobacterium tuberculosis or tuberculosis is only shows up in internal organs. Mycobacterium leprae, which causes leprosy, only has effects on the extremities. Why might this be the case? It's a good guess. It has nothing to do with oxygen, but that's a great guess. It has to do with temperature. Yes, the cooler regions of the body are colonized by the causative agent of leprosy. Its temperature preference is only a couple of degrees different than Mycobacterium tuberculosis, but that's enough to discourage the leprosy organism from colonizing deeper into the body where it's hotter. And it's also enough to keep the Mycobacterium tuberculosis from colonizing the extremities where it's cooler. So the temperature differential is very little. Mycobacterium leprae is adapted to 35 and tuberculosis to 37. But that 2-degree difference is enough to restrict where they can grow in your body. Typically, are hypothermophiles or psychrophiles human pathogens? Let's just start with this, yes or no. Great. Why not? Everyone said no, and that is correct. And some said it was either if it's hypothermophiles, our bodies are too cold. Psychrophiles, our bodies are too warm. Let's think. So given that that's a sort of a definitional answer, right? By definition, hypothermophiles don't grow at 37 because it's too cold for them. And psychrophiles don't grow at 37 because it's too hot for them. What's actually happening in these organisms when they are in the human body? Let's start with hypothermophiles. What happens to hypothermophiles in the human body? Why are they dead? And note this instruction. Go beyond the surface or circular answer. Go beyond a definitional answer. Go to a mechanistic answer. Yes, Carlo. So some of you are saying, which is true, they can't survive at those temperatures. That's a definitional answer. It's accurate, but it's not enough. So let's think about what that temperature is doing to them. Carlo has said their membranes are either too rigid or too leaky. So that's true. The membranes are going to be affected. Who's going to have, of these two that are dead in the human body, which one's going to have a membrane that's too rigid or too leaky? And what else is happening? They can't perform cellular functions anymore. Why not? their proteins are denatured? Well, only in one case, okay? The proteins are going to be denatured for one of these guys. Which one? Remember that being too hot denatures proteins, but being too cold does not. So who is in the human body? Which one of these guys is going to feel like it's too hot? That's right. The psychrophiles proteins are denatured. What about the hyperthermophiles? What's wrong with their proteins? They're in a situation where they think it's too cold. So their proteins have done what? What's happened to them? They're frozen. That's exactly right. Their proteins have become too rigid and can't function. Okay. All right, let's move on to pH. Okay, there's 3 categories of organisms by pH. We can see that some of them like this, like low pH, some like medium, some like high. So what do we call it when they like these low pH conditions? That's right. We call them, low pH is acidic. We call them acidophiles. When they like these pHs around 7, what is this? What is it called? This is a neutral pH. These are called neutrophiles. And then the high pHs, it's also called a basic pH or an alkaline pH. That's right, alkalophiles. Good job. Now, if we look at temperature, there's lots of examples of microbes in all of the different categories of temperature. That's not the case for pHs. There's examples of organisms in all of these categories, but the preponderance of the majority of microbes actually only fall into one of these categories. So which one is it? Most microbes belong to which of these 3 categories? That's right, it is neutrophiles. Now, when we think about why pH matters, what structure or things inside the cell are affected by pH? What does pH affect? Well, the right pH will support proper function of something. The wrong pH will do what? What is affected by pH? If you're not sure, you can put a question mark, but make sure to give an answer. It's enzymes again, Carlo. You are right. It's all proteins, really. Why? Because pH affects hydrogen bonding. Proteins are held together by hydrogen bonds. You know what else is? RNA. Now you might think, mRNA, who cares about that? But there's other kinds of RNA, as we'll remind ourselves in the genetic section, things like tRNA and rRNA that have 3-dimensional structure, and pH will disrupt that and cause it to not work. And so what we can see here is, interestingly, the graph's symmetrical, unlike temperature. And that's because whatever's happening at the wrong pH on the high pH side is the same as what's happening on the low pH side. So that tells us that it's really, as we deviate from this optimal pH, we're getting denaturation either way. It's not like temperature where you get denaturation on the hot side, but then you just get slowing down on the cold side. Okay, so their next topic is osmotic pressure. Okay, and there's quite a bit to say about this, so I just want to take a quick check-in. We could take our break now, or we could move forward and sort of take the break at a different time where it conceptually makes more sense. So do you want to break now, or do you want to break a little bit later? Let's have a vote. Okay, so you want to keep going. All right, let's do it. Soldier on, good folks. Now, when it comes to water, osmotic pressure, it's a little bit tricky because it's really not so much the water itself as what's in water that matters. Water will travel across membranes with no problem. It chases whatever's dissolved in the water that water likes, like salts, sugars, and ions. So when we talk about something like tonicity, what we're referring to is what's the relative concentration of stuff outside the cell versus inside that water likes. what percentage of salt is considered to be isotonic for most cells? That would be where the cell's insides are at a particular percentage of salt, sugar, and ions, and the outside world is similar. So overall, the net movement of water is 0. What's that percentage of salinity that's considered to be isotonic for most cells? We, of course, are included in this. Yeah, it is around 1%. Humans, it's like 0.9. Okay, so it's around 1%. If human cells are in a hypotonic situation, what happens to them? They explode, correct. Hypotonic, the world is watery. That means water is going to rush into the cell to dilute the interior of the cell, and we do not have anything to prevent that from happening, so we'll explode. Bacteria typically don't explode because of cell walls. What would make them vulnerable to hypotonic conditions is if we were to remove that cell wall. Can we think of anything that would damage a cell wall? Yeah, some antibiotics. Not all antibiotics, but some of them, okay? In particular, penicillin against gram-positive doesn't work against gram-negative. So age or antibiotics, something that will degrade the cell wall. So antibiotics can do it. Also, as cells age, just like us, they start to fall apart. When you take food and you add a lot of salt to it, it typically does not spoil as quickly. Why is that the case? Why aren't the microbes that might land on that meat not doing anything? Yes, that's right. Because the high salt condition is considered hypertonic for cells, and a lot of them are just going to be dehydrated. And bacteria are not usually protected from that. There are some exceptions, but typically the cell wall does not protect them, certainly. Now, some organisms are so adapted to high salt environment that they actually have to have it or they're dead. What do we call them in that case? What's their category? Yeah. So yes, halophiles is part of it. So loving salt, halo is a word that means salt. Halo, halogen from chemistry. Halophile, salt loving, and because they require it, they're called obligate halophiles. Obligate halophiles. Now, there are also organisms that are halotolerant. This is a word that means they can tolerate high salt. They actually prefer normal tonicity, like, you know, that a human cell would be happy at, but they have ways to handle high salt. What human organ is heavily colonized by halotolerant microbes? So think about what part of your body is heavily colonized by organisms that would be halotolerant, and why would they need to be? Okay, what part of your body would be colonized by things that can survive in high salt? That's right, Jolene. Indeed, Alyssa. Yes, Sydney, it is your skin, because we sweat, right? And sweat contains salt. And what genus is famously halotolerant that is a common resident of human skin? That's right, it is Staphylococcus, indeed. Okay, so moving on, we're going to talk about oxygen. And this is really important for those of you who are ready to move on to part 2, which should be everyone, and have the starting part of your dichotomous key says what's the oxygen requirement or oxygen preference for your organism. So we're going to talk about this, and we're also talk about a test that is involved. The effect of oxygen on growth is complicated. This is because some organisms, not all, some like us, need oxygen. Why do we need oxygen? What do we use it for? That's right, Aubrey. Indeed, Mackenzie. Yes, Jolene. Yes, Christina. We use it for aerobic respiration, where we get the majority of our energy. Without it, we were dead. Okay, that's our metabolic strategy. Now, some microbes don't need oxygen. Why not? What do they do instead? They're clearly not doing aerobic respiration because, or let me say that differently. They don't rely solely on anaerobic respiration. Some of them can, some of them can't, but they don't need it absolutely. So what do they do instead? If there's no oxygen, how come they're alive? Yes, they can do some kind of anaerobic metabolism. Okay, notice that I used the word metabolism, not respiration. That's on purpose. This is a category of metabolism that includes multiple different processes, including anaerobic respiration, but also fermentation, which is not the same thing. Okay, now this is a separate question. Why do some microbes die in oxygen? There are some organisms that not only don't need oxygen, but actually can't tolerate oxygen. So what's the difference between the ones that can live in oxygen and ones that can't? What is it about living in oxygen that's sort of like risky, or how do you need some? Great. If you're not sure, putting a question mark is an excellent choice. Yes, Aubrey, oxygen can be poisonous. And if you're going to live in oxygen, even if it's a boon, even if it's useful for this aerobic respiration, it still can be poisonous. And if you're going to survive, you're going to need some protections. Why is it poisonous? Mainly it's poisonous because oxygen has a tendency, because it's highly electronegative, it has a tendency to steal electrons and become slightly negatively charged. And that molecule, oxygen that's not neutral but slightly negatively charged, is very reactive. It runs around in the body, bumping into membranes and proteins and carbohydrates and damaging them. Okay, so it's one of these reactive oxygen species that cause premature aging and damage. High enough level will kill you. Organisms that can grow in oxygen have defenses to detoxify the toxic byproducts of living in oxygen. Okay, so if you want to know where microbes can grow, whether they need oxygen, whether they can grow without it, if they're killed by it, all these different questions, you can ask that by putting them into a special test called a thioglycolate agar test. How it works is that this is an agar, it's a solid food that has been depleted of oxygen, and then the oxygen diffuses in from the top. So it's a gradient. Here, it's full oxygen, normal air. Down here, there's no oxygen. In the middle, there's a medium amount. Then you just put your bugs in all the way through and see where they grow. Okay. And here is basically the growth pattern of the different categories of organisms. So the first thing we have to know is, what are all the categories? So let's start. I'm going to call out a letter and you're going to write into the chat what that category is called. So here we see guys growing only at the top of the thioglycolate agar. What do we call these guys? They're clearly growing in oxygen, but not without it. So what's our category? Good. That's right. This is an obligate aerobe. Obligate aerobe. Okay, so it's growing in oxygen, not growing without it. It has the ability to use oxygen in its metabolism, and it doesn't have any backup strategy. All right, let's go to this one. Here we can see an organism that's growing at the top and at the bottom, but we do notice it's growing better at the top than at the bottom. What's this category called? If you don't remember, you can put a question mark, or you can look at the chat and see what other people are saying and go, oh yeah, I remember that. Let me put that in real quick. That's right. Facultative anaerobe. Facultative anaerobe. Able to grow without oxygen, capable, but really would prefer if there was some oxygen. Over here, we've got this guy. What do we call this guy? Yeah. Obligate anaerobe, only can grow with no oxygen. Oxygen obviously is killing it, right? But it's able to do something else. Now, interestingly, over here, we see a guy that's growing all the way through the tube. There's really no difference between the top and the bottom. Okay, so what do we call these? That's right. Aerotolerant. So it's growing using some metabolic strategy that does not rely on oxygen. And it basically just has some way to defend itself against reactive oxygen species, but doesn't actually like use or care about oxygen particularly. And last but not least, what's this guy? That's right. These are called microaerophiles. These are actually obligate aerobes, but they can't tolerate high levels of oxygen. They need it, otherwise they'd be alive here. Since they're not alive here, we know that they need it. But they don't have very strong defenses against oxygen, so they kind of need a low concentration. Okay, so here's the names and definitions of all the things. Just going to check our time, make sure I'm not like dragging you through the mud too hard. So let's just revisit what's complicated about oxygen. So we need it, and some microbes need it for aerobic cellular respiration. And the reason that when you look at this tube, for example, let me go back here. Okay, you see this guy, he can live with oxygen, he can live without oxygen. But when he's living in oxygen, he's actually using it in his metabolism. And you see that the growth is faster, thicker, right? That's because when you do aerobic respiration, you get the most ATP. Of all the different possible strategies, metabolic strategies that organisms can have, really the best one is aerobic respiration. It gives you the most ATP per glucose if you're able to do it. Now, the downside is you are definitely creating a lot of poisonous reactive oxygen species because we're constantly loading up oxygen with electrons at the end of the electron transport chain. And if you don't have some way to detoxify that molecule, you are in deep trouble. And so you're going to generate a lot of these guys. This happens anyway. You're just in an oxygenated environment, even if you're not doing anything, like aerotolerants aren't doing anything with oxygen, but they're around oxygen molecules. And some of those oxygen molecules have a little extra electron and they're running around bumping into things. But if you're actually using oxygen in your metabolism, you're creating a buttload of this. So how do you protect yourself? If you're going to survive in oxygen, whether you're using it or not, you're going to need some enzymes. So organisms that are growing oxygen have something called superoxide dismutase. It takes any superoxide anion, oxygen with a little bit of extra electron on it, and converts it to regular oxygen. So if you're growing in oxygen, you have this for sure, 100%, because this is the most dangerous one. If you're actively using oxygen and creating a lot of this, it's converting it to hydrogen peroxide, which is still bad. So you're going to have another enzyme. Okay, so pretty much what that means is that obligate aerobes, microaerophiles, and facultative anaerobes all have superoxide dismutase plus one of these enzymes. And aerotolerants have just superoxide dismutase, and that's enough to protect them. And obligate anaerobes don't have any. Okay. So let's just check in and see if we can translate this information, sort of apply it a little bit. Who's got defenses against reactive oxygen species? Put your letters into the chat. Who's got defenses against reactive oxygen species? Put your letters into the chat. Any defenses? If you're not sure, you can put a question mark. That's a totally legit answer. But when you look at these guys, who do you know has got some kind of enzymes to defend themselves against reactive oxygen species? Yes. The good answer is all of them except for C. Okay, so he has no defenses. Everybody else has got defenses. Why? Well, this is the only one that's dead in the presence of oxygen. Now, which of these organisms actively use oxygen in their metabolism? Not just able to survive, all of these guys can survive, but who's actively using it to do aerobic respiration? All right, good. So most of you picked A, which is accurate, but there's more than that. We can also know that B is using oxygen actively, at least at the top, and so are microaerophiles. Okay, and we know that they are using oxygen because if they didn't need it, they would be alive down here, but they're not. How do we know these guys are using it? Well, they're growing faster at the top, which means they're using oxygen. Who needs oxygen to survive is clearly A, but also D. Again, anybody who's not growing at the bottom. Now, when it comes to who's got which defenses, this is a little bit something you have to memorize. I can give you a reason, maybe the logic will help you remember, but you kind of just have to remember it. So remember that the defenses are superoxide dismutase. And if you live in oxygen, you've definitely got this enzyme. And then either catalase or peroxidase in some cases. And that's only going to be if you're actively using oxygen. So we know that if you're alive in oxygen, you've got superoxide dismutase. That's going to be this guy, this guy, this guy, and this guy. And that you additionally have catalase or peroxidase if you're actively using oxygen in your metabolism, which would be this guy, and this guy. Okay. Now let's take another close look at this. When we look at the microaerophiles and the obligate aerobes, there's similarities and differences. One similarity is they can't grow without oxygen. So what kind of metabolism must they be doing? They're both doing what? What do they have in common in terms of their metabolic strategy, which is their only strategy? They both require oxygen. What are they doing with it? That's right. They're doing aerobic cellular respiration with it. Now, the difference is this guy can grow in full oxygen. This guy can only grow in partial oxygen. And so he has a lower amount of superoxide dismutase in either catalase or peroxidase. Let's take another comparison. Let's look at the difference between this facultative anaerobe and this aerotolerant guy. Both of them are growing through the whole tube. But what's different is that this is growing better. How come? What can this guy do that this guy can't? Yes, that's right, Sam. The facultative anaerobe, when oxygen is presented, is able to use it for anaerobic respiration, which remember gives you the most ATP per glucose. And then he has some kind of backup strategy. When the oxygen goes away, he switches to something else, okay? Whereas this guy has one play. He does one thing that does not require oxygen. And when oxygen is around, he has protections against it, but does not actively use it. So does not do aerobic respiration, and therefore is not able to get this extra boost in oxygen because he can't do the more efficient thing. All right, so quick poll, just write in the chat really quickly. From this whole page, you understood this yes, meaning completely, no, not at all, or kinda. Some of the things made sense, some of them didn't. Understood it yes, no, or kinda. Excellent. Okay, so let's move on. Good job. So this is just a little summary of who's got what, just so you can use that in your study practice. Okay, so we are going to take our break now because you have hung in with me long enough. Well, maybe I'll just go another 3 minutes and then we'll start at noon. That'll be a nice round number. So we're going to do just a little quick application exercise. So I'm going to tell you about Staphylococcus aureus. Again, you don't have to memorize these facts, but I'm going to give them to you and you're going to interpret them. So one thing about Staphylococcus aureus, which is a common resident of human skin, is that it's able to do aerobic cellular respiration, and it's also able to do an anaerobic metabolism called fermentation. Based on this information, what category would you put it in? What kind of like category is this sort of like definition of? We've talked about all kinds of things, right? We categorize microbes in lots of ways. What's one term we can now use to describe Staphylococcus aureus based on this information? Facultative anaerobe. Good job, Mackenzie. Yes, Christina. Okay. Facultative anaerobe. This is describing its oxygen preference / utilization / survival. So we can say confidently that's a facultative anaerobe. Oxygen's around, it's able to use it. Oxygen's not around, it's able to do something else. Based on the fact that it prefers neutral conditions, what category would you assign it? That's right. It likes pH to be neutral, so we call it a neutrophile. Neutrophile. If you learned that it grows the very best at 37 degrees, what category would you assign it based on this information? That's right, it is a mesophile. And if you learned that it prefers 1% tonicity, but can tolerate up to 10% salt, how would you describe it then? What word would you use to describe it? It's halo something. Yes, Jolene, that's right, Carlo. It is halo tolerant, halo tolerant. And last but not least, knowing that it can cause human disease makes it a pathogen. So we can see all these different categories can now be used to describe a real thing. Okay, so when we come back from our break at the top of the hour at noon, we're going to talk about food, other conditions, and move on to other parts of this topic. But for now, go take a well-deserved break. All right, welcome back, everyone. Let's continue our conversation about growth. So one of the other important things that you have to pay attention to, that's very important, obviously, is what you're feeding organisms. And they need all kinds of things. They need a carbon source. Depending on the type of organism, that's either going to be organic or carbon dioxide. So they either need like, sugars or proteins or lipids, or they need CO2 as their carbon source. And they're going to need some kind of nitrogen. Usually that comes from proteins, because the amino group on the protein. They need a source of sulfur, usually also found in protein. They need some kind of phosphorus, usually from the environment, and trace elements. One concept or term to be aware of is growth factors. Growth factors are basically any specific nutrients that have to be added to the media that cannot be made from basic ingredients. So things like particular sugars or particular amino acids, things like that. Now, if an organism has a lot of growth factors that it needs, then it's termed fastidious. Fastidious is a word that means picky. So if it needs a lot of very particular things and it doesn't have the ability to make most of the complex chemicals itself from just, basic ingredients, then it would be termed fastidious. And how we empirically determine if it's fastidious is it's defined as an organism that has so many specific growth requirements that when you put it on a rich medium, like the TSA foods that we've been using in lab are rich, it doesn't grow. So when we think about how we sampled the environment in lab 1 and then what we looked at in lab 2, do you think that you grew everything that was out there? Let's say you swabbed the floor, okay? And you ran that over the surface of the TSA. Did you grow everything that you captured with your swab? Excellent. Healthy skepticism. The answer is probably no. There's bound to be something that you picked up that was alive or potentially could have grown, but TSA wasn't the right medium. The 37 degree wasn't the right temperature, or 25, whatever it was. The food that's in the TSA was not the right food. The salinity was off. Maybe it doesn't grow in oxygen, all those things. Speaking of media, there's several different ways we can feed microbes. So we have some kind of recipe. We can make that into a soup and pour it into a tube. That's a broth. Or we can add a solidifying agent called agar and pour it into a dish, or again into a tube or into a slant. And they all have different purposes, but they're all different ways to make a recipe and that some cells will be able to eat. There's a bunch of terminology we need to know, so we're just going to do really quick matching and then fill in the blank. So here are your options for terms, and I'm going to provide a definition, and you're going to tell me which of these terms this is best defined by this. Several species mixed together. Yes, mixed culture. When you take your loop, as you did for the T-streak, or as you will, as you did for the T-streak, when you pick up your loop to transfer onto a new medium, what is actually on your loop? What's it called? Yes, that's an inoculum. And then you put it onto new food that doesn't have any microbes on it already. And that new food, before you've added it to the new food, is considered to be a sterile medium, right? Sterile medium. Now, the act of transferring that inoculum to the sterile medium is what? Indeed, it is an inoculation. And then when we grow it for some period of time in some particular conditions, what are we doing? Correct. We're incubating it. Now, these isolated colonies that we're going to see on the T-streak this week all derive from a single original cell. And if we're able to create a situation where everything in a medium comes from one species and originally came from just one cell, then that would be the definition of a pure culture. Now, it is important to recognize that we may not get a pure culture in the first step after doing a T-streak. We'll find out this week how we did, because, you know, yeah, if this is an isolated cell and it grows into one colony and we take this and we make a plate out of it, and everybody here is all the same species, well, that's a pure culture. But if we took our loop and sampled this region right here, you see that there's two colonies right next to each other. Does everyone see that? And if you take these two colonies and you made a T-streak, well, then you're going to have a mixed culture still, because both species will still be there. Okay, now we're going to do a really quick fill in the blank. A cotton swab is used to sample the school urinals, spread on a TSA plate, which is then wet at 25 degrees for 2 days. Incubated. The environmental sample plate here is an example of a correct mixed culture. A sterile loop is then used to transfer some blank from the environmental sample plate and transfer it to a new sterile TSA. Inoculum. What method is performed to isolate colonies? Correct. It is a T-streak. A plate of isolated colonies, if they're all the same as each other, is considered to be a pure culture. Okay, great. Now, as I mentioned earlier, there's lots of different recipes for media. There's two categories, complex and chemically defined. So let's look at that, right? Here is a recipe that contains pure amounts of carefully measured, precisely measured pure chemicals. So there's a precisely measured amount of pure glucose, precisely measured amount of different salts, and then some water. Is this considered to be a complex or chemically defined medium? And if you're not sure, putting a question mark is perfectly good. Okay, this is considered chemically defined. Chemically defined is we have precisely measured amounts of pure chemicals. We know exactly to the molecule how many, of theoretically at least, how much of everything there is. Here's another recipe. It's got water, it's got measured amounts of agar and salt, but then it's got something called peptone, which is a partial digest of protein sources, and beef extract. This clearly has a lot of proteins in it, and it is got a lot of stuff. It's considered rich, but we don't actually have a precise accounting of exactly which amino acids and which ratios are present, or what other things might have come in this beef extract. So we would consider this to be complex or chemically defined. Yeah, this is an example of a complex medium. Anytime you see peptone, which is a partial digest of a protein source, or beef extract or extract of anything, you know we're dealing with a complex medium. So here is a very typical 2 types of media that you'll see in lab. We've already used this one. The one that we've been using in lab so far is called TSA, tryptic soy agar. And we also often use something called glucose salts agar, so TSA and GSA. TSA has casein digest, so casein is a milk protein, so a digest of milk proteins. Digest of soybeans, so good source of protein, some salt, and some agar. Here, there are precisely measured amounts of glucose and various salts, glucose salts agar. Which of these would we use if we wanted to sample the environment for unknown bacteria? Would we use either one? It's just fine. Would we use the TSA or the GSA preferentially? Great. Everyone is saying TSA, and that is what I would use as well. Raise your hand if you can explain why. Why would you use the TSA over the GSA? Raise your hand if you can explain why you made the choice you made. Spencer, go ahead.
It's just it's broader. Like there's more stuff in there that the bacteria can use.
Yeah.
Potentially.
Yes. So remember we talked about fastidious organisms that wouldn't even be able to grow on TSA. But lots of organisms do have some growth factors, like they need some number of amino acids. They can't just make them from sugar. As an example, okay, we are like this. We are not fastidious, but we do need certain complex organic compounds to be supplied to us. There are some amino acids that we can't make from scratch. We actually have to have them in our diet, such as tryptophan, as an example. Whereas some microbes are capable of making all the amino acids, right? So E. coli, for example, can grow on this. You give it glucose and some salts, and it's able to make everything that it needs from that starting material. But we're not like that. And lots of microbes are not like that. So this is a better choice because we don't really know ahead of time what the nutritional requirements are for the microbes that we're trying to sample in an environment. So the more broad our medium can be, the more stuff that it offers, so that a microbe doesn't have to make everything from scratch, the better. All right. This week, we're going to use more than just TSA. We're going to use selective and differential media. These are useful for identifying and testing species that have already been purified. Selective media means that this food inhibits or kills certain species and encourages or allows to grow certain other species. Okay, so it's a dead or alive assay. You have a pure culture, you want to ask a question like, can you tolerate high salt? Put it on a high salt plate, does it grow or not grow? Then the answer is yes, or it is halotolerant, or no, it's not. Or you want to ask, is it, can it survive high pH? And you put it on a high pH plate, right? And then it's either alive or it's dead. So that's selective media. Differential media, on the other hand, is media that may grow lots of different things, but has some kind of substance that microbes change in a visibly identifiable way. And so you can then say, oh, well, these guys all grew, but these guys are purple. So that tells us that they are, able to ferment a sugar. And these guys are green, which means they can't, or something like that. We're going to start with an example of selective media that we will use in lab this week. and that you do have to know for the test, and it is called PEA. That stands for phenylethyl alcohol agar. It is selective for gram positives. Okay, so what we've done here is we've grown two species, E. coli and Staph aureus, on a TSA plate, which is not selective. This grows everything, or grows both these species pretty well, and on a PEA plate, which is selective for gram positives. Okay, it should encourage the growth of gram positives, and it should kill things that are not gram positive. So looking at these data, the PEA plate versus the TSA plate, what do you conclude? Write that into the chat. What is this data telling you? If you have no idea, you can use a question mark, but you can also read the chat and see what other people are saying and see if what they're saying makes sense to you. That's right. What this data tells us is that Staphylococcus aureus is gram positive. How do we know that? Well, it grew, and E. coli did not. Now, one thing that we need to ask ourselves is, why did we do this control? This is TSA, it is a control. Why was it important? When we look at this plate, we can see that Staph aureus grows really well. So it's pretty easy to say, oh, it's gram positive. But E. coli, you can still see where it was plated, and it's kind of hard to interpret this. Maybe this could tell us that E. coli is just a slow grower. How do we know that it's not? What was helpful about doing this plate? That's right, Sam. The control shows us how it grows when it's not in a selective environment. It grows just as well as Staph aureus if it's not being challenged. So when we look at this, we can see, oh no, it's not a slow grower, it's sad. And so it's probably being killed by this plate. Does that make sense? Yes, no, or kinda? Great. So in lab this week, we'll get a chance to do this test, and you'll find out what things are gram positive and what are not, and you'll have a control just like this to compare to. Here's another plate. MSA stands for mannitol salt agar. As you might imagine, there's salt in there, 7.5% to be precise, and a sugar called mannitol. There's also a pH indicator dye, and this dye is red at neutral pH and yellow at acid pH. Why that matters is that some organisms take sugar and ferment them. Mannitol fermentation is one way to distinguish organisms from each other. And fermentation produces acid as a byproduct. Now, when we look at the amount of salt in this plate, what do we conclude? This condition would be what for most microbes? Not all, but most microbes would find 7.5% salt to be what? Correct. Most microbes would find 7.5% salt to be hypertonic, and they would be killed. So this plate is going to select for ones that are what? It's going to select for what category of organism? Think about what we learned, some of those terms we learned earlier today, or reviewed since you learned them initially in doing your prep for this class. That's right, halotolerant. Can they tolerate high salt? Okay. Now, this plate is useful because it kills most species, but not Staphylococci because they're halotolerant. And Staphylococci can often be distinguished from each other, or at least sort of subcategorized based on whether or not they do or do not ferment mannitol. And there's a pH indicator dye in here, right? That I told you earlier was a particular color at neutral pH and a different color at acid pH. So what is this plate selective for? That's right. It's selective for salt tolerance or halotolerance. Now, we've talked about species that are salt tolerant, right? Staphylococci are typically salt tolerant. Look at this plate and see if that's what you see, Okay, so we've got E. coli didn't grow, Staph epi and Staph aureus grew, and this one's yellow. So which species are salt tolerant based on this plate? Yes. Because they grew, and the growth is what we're looking at to answer this question, because they grew, both Staphylococci, Staph aureus, Staph epi, they're both salt tolerant. Which one of these two species ferments mannitol? That's right, it was Staph aureus. How do we know? Because the agar turned yellow, which means that the pH indicator dye is showing us an acid pH, which would be because the mannitol got fermented. Let me ask you this question. Think carefully. Does E. coli ferment mannitol? If you're feeling uneasy answering this question, you should. Okay, the answer is actually we don't know. Some of you are kind of there, but let's do a raise your hand. Why is the answer we don't know? Not yes or no, not even maybe, but we don't know. Well, maybe I guess would be the right answer. Sam, go ahead.
Oh, because it didn't grow.
Yeah, it didn't grow. Because it didn't grow, we can't ask it any questions. We basically, we're trying to interrogate a subject. Well, we killed them before we'd ask if he was there on the night of August 19th. So if you have a selective and differential plate and the organism dies on the first question, which is, are you salt tolerant? You cannot follow that up with a, well, I see you're not salt tolerant, but can you ferment mannitol? Because he's already dead. He can't answer that question. All right, this is a final example of a differential medium, just to help us understand that it's not all about like yellow versus red, although this appears yellow and red. It's not all about color, but sometimes it's about clearing. So some media are opaque. And this is an example. This is a blood agar plate. It's opaque because actually there's red blood cells in here, and they block the light so you can't see through the plate. 3 different species have been grown on a blood agar plate. And what happens is either they can or cannot explode red blood cells to use them for energy. If they can, they're going to clear the blood and you'll be able to see through it. Again, this is a differential assay. It's not a dead or alive. All 3 species are alive. It's just, what does it look like? Okay. And so this is a useful test sometimes to help us detect pathogens. Some pathogens explode red blood cells and get energy that way. All right. We have to talk a little bit about inoculations. We started this conversation last week, just a smidge, and we're going to revisit it this week in lab. But when we inoculate plates, there's 2 major methods. We can do a T-streak or a single streak. What was the goal of the T-streak method? Again, write that into the chat. The goal of the T-streak method. That's right. Is to isolate colonies, either to purify a culture from a mixed culture or to maintain a pure stock that we already have. Either way, but we're trying to produce isolated colonies. In the single streak technique, we just take an inoculum of an already pure culture and we just streak it down. We're not trying to get single colonies. We're going to cause a line to grow. We don't care about individual colonies because we're assuming that our starting material is pure. And so which technique would purify mixed cultures to pure cultures? Well, that's obviously the T-streak, right? Which technique would make most sense to use when you already have a pure culture and you just want to ask it questions? Correct. If you already have a pure culture, there's no reason to do isolation. Might as well just make a single streak. And so you're going to do that a lot this week. Okay, we're not taking our break. We already had our break, so let's move on. We're going to talk now about how we measure growth, and then we'll see if we have time for the math. When you're growing something in a lab, it's useful to be able to look at it and see if you did it right. So if anything's there. In your background or in the prep that you did, there's several direct and several indirect methods that you have to know about. And in the world, there's many, many more than just these four. So listed on this page are the ones you need to know for the test. Listed in red are the ones we're going to go over today. Okay, but you do have to know what direct counts are and what dry weight is. So for the direct method, we're going to look at dilution plate counts, and for the indirect method, we're going to look at turbidity. Direct methods of growth are when you actually are counting cells or colonies. So you're getting a precise count of what actually is there. This type of measure works if you have something that actually forms colonies. Not all things do. The way that this works is very similar to something you've already done. In lab 1, do you remember that you did CFUs per mil to detect the amount of stuff that can grow in a water sample? So now all we have is a broth of some microbe we tried to grow in lab, and we're just trying to find it. We just want to know its concentration or how many cells there are. So what we could do is take a milliliter of that thing and put it on a plate, but it's probably millions of cells per milliliter. So if we put 1 million cells on a plate, it would all grow together and form what we call a lawn. So what we do instead is do dilutions. If we did a 1 in 10 dilution, let's say we had 1 million per mil to start with, we're still going to have like 100,000. And when we put them on this plate, it's going to grow into craziness. Even a 1 in 100 or 1 in 1,000 dilution is still going to give us cells that are stuck together. It's only as we get into these more dilute concentrations that we start to get individual colonies, which is what we need in order to count. Now, of the options here, which one plate would be best to use if we're trying to do our CFUs per mil? Well, plates, you can identify them by the dilution, which is either 1 in 10, 1 in 100, 1 in 1,000, 1 in 10,000, or 1 in 1 million. So which one would you choose? Great. And just think about why you're making that choice. You know, just have that in your mind. Excellent. So I would say it's a pretty even split in the class between the 1 in 10,000 and the 1 in 100,000. I'm just going to ask a couple of you, I'm going to cold call a couple of you to give me your reasons. And, you know, it could be anything. Like you could just be like, I guessed, or I do have a reason and here it is. And you don't have to be right. I just want to hear what they are. So let's start with Esther. Esther, you said 1 in 10,000. What was the reason for choosing that one?
Because we have like a number of them, so like they're countable.
They're countable. Okay, great. Jolene, you chose 1 in 100,000. What was your reason?
Same reason. Like it looks like it's the easiest to count, but I was kind of unsure as well. Yeah,
excellent. Okay, so these two do make sense in terms of being possible to get an actual number. These are all impossible to count, so these would be terrible choices. Of these two, however, the better one is this one. And the reason for that is it's a countable number, but there's more of them. And the more dilute you get, the more you are at the mercy of random variations due to just random chance, right? Every time you do this dilution and you put some cells on a plate, do you think you get the same exact number every time? No. Right? There's going to be a little bit of variation because the cells are all, you know, floating around, and then some, you might just by chance, you might get a few more cells here versus that other sample. The more dilute it is, the more those small changes make a big difference in the final answer. So you're going to get a more accurate answer the more you actually have. Yeah, outliers make this, like you could accidentally have gotten, not accidentally, just by chance, you might get an extra 2 cells here. That's going to change your final number by 50%. If you get an extra couple cells here, it's really probably only going to change your number by like 10%, something like that, 10% probably. So it's a big difference. The other thing is that we always want to go for, if possible, somewhere in this range. At least 30 gives us a better statistically significant answer. And around 300 is the limit of what you can really count accurately. Okay, so if this is the one we're choosing, I'd like you to use the math you already know how to do, right? Look at what the volume is, look at what the dilution is, look at what the colonies are. What was the original concentration of the broth that had these cells in it? Write that into the chat. If you want to put your math in, that would be even better. I'll give you a second. Just waiting for our last few answers to come on in. Yeah. Always make sure to report your answers with units. In this case, the units are going to be CFUs per mil. So I counted 32. Many of you counted 34. That's completely fine. But whatever that number of colonies was, times the dilution factor of 10,000 divided by the volume, which is 1 milliliter, gives us 320,000 CFUs per mil. Now, if I had gotten this data, but in my experiment, I'd actually used 0.1 mils, what would be the new concentration? What would the answer have been in this case? I'm not saying that I'm just saying if we'd gotten this and the experimental parameters were actually different, what would our answer be in this case? Put that answer into the chat. Whatever your number that you got, 32 or 34, you have to multiply it by the dilution. You have to divide by the volume. Great. So in this case, when we go to divide by volume, we'd be dividing by 0.1, not by 1. And so that would increase our number by 10-fold. So 3 point something times 10 to the 6, or 3 million and change CFUs per mil. Okay, let's move on to turbidity. So turbidity, another way to say it is cloudiness or opaqueness. We can measure this using a spectrophotometer. And what you can see in the way that it works is we shine a light through the broth and we detect how much light comes through on the other side, and this will show us a reading. When there's not very many cells, there's lots of light that goes through, so we get a high light reading. And when there's lots of cells, more concentrated, not very much light comes through. So as a broth gets more concentrated, more or less light reaches the detector. That's right. The more cells there are, the less light will reach the detector. So which tube is shown by these readings? So tube A, if both these are measured and both those measurements are shown here, which one should belong to tube A? Great. That's right. The more concentrated something is, the more turbid or opaque or cloudy it is, the more growth there is, the less light will reach the detector. So a low light reading tells us more cells per mil. Okay, we still have a little bit of time for math. I'm sure you're so excited to hear that. So we have a couple of different things we need to talk about. I want to just revisit this colony idea. The colonies come from an individual cell that's just given enough time to divide many times and produce a clump of cells that are basically a clone of each other. Now, I want to remind you that bacteria by themselves, the first cell that lands on there is invisible. It's so small. By the time it's divided a number of times and formed a colony, it's now visible. Basically, it's 1,000 times bigger, okay? but we've already talked about how it's not the 1,000 cells, it's more like 1,000,000 cells at least. And this took not very much time, okay? One cell to 1,000,000 takes 1 or 2 days. My new question is, how many cell divisions were involved? How many cell divisions were involved? So thinking about this, so 1 cell divided to produce 2, so that's the first cell division. 2 cells divide to produce 4, that's the second cell division. 4 becomes 8, that's the third, okay? 8 becomes 16, so it's exponentially increasing. How many cell divisions or how many generations are required to go from 1 to 1,000,000? Put your answer into the chat. I should say put your guess into the chat. No abstaining. Take a risk, throw those dice, throw a number out, even if it's a random number. How many cell divisions do you think does it take to go from 1 to 1,000,000? Great. So we have a range of guesses in the class between 20 and 5,000. That's a pretty big range, right? Now, as it happens, we can actually find out. If we don't have to guess, we can find out with some math. And that is because, as I mentioned, the number of generations is predictable in that when 1 cell divides, it becomes 2, and 2 cells divide become 4. This period of time, or this is 1 generation. 2 to 4 is 1 generation. 4 to 8 is 1 generation. 8 to 16. This is known as exponential growth, where the population doubles every time there's a cell division. And this is happening predictably as long as the cells are in a pretty stable, consistent environment that they like. So generation time is, in a particular set of conditions, how long does it take the population to double, right? The amount of time it takes to go from 1 to 2 is the same amount of time it takes to go from 8 to 16. This is 1 generation, and then when it divides again, that's again another generation. That will take the same amount of time. Is the generation time the same for all species, do you think? Yeah, the answer is obviously no. Obviously no. We already can think, well, human generation time versus a E. coli generation time, but even within bacteria, it's not always the same. And the reasons for that are many, but now that we've gone through all this stuff about different conditions that things like, and we take 3 different species and we put them all in the same conditions, some of them are going to grow faster than others because those conditions are better for that particular species. Or maybe that some of them can't grow at all. And then we also talked a little bit about different metabolic strategies, right? What metabolic strategy gives you the most ATP per glucose? What was the most efficient metabolism? Gives you the most energy? Aerobic respiration. Do all species do aerobic respiration? No, some of them don't. Some of them rely on fermentation, which is super slow. So there's all kinds of reasons, but it comes down to what metabolism can they do? What conditions can they tolerate? And that's all about their genes, basically. Let's do a sort of concrete example. Like, let's take a mesophile and put him in 2 conditions where he's able to grow, right? Let's put him in the refrigerator. Let's put him inside your body. Where is the generation time going to be the shortest? That's right. It's going to grow faster in your body. The shortest generation time is going to be in your body. All right. So now let's proceed to the math. There is an equation, and maybe you're an equation person, so here it is. And if you're not an equation person, don't worry, I'm going to give you another way to think about this. But let's just lay it out. Okay, so let's suppose that we have any two of these variables, we can calculate the other one. This is a number of cells in a population after a particular thing has happened, and this is the number we start with. And right here is the number of times the cell divided. So let's say we start with one cell and we allow it to divide a number of times. We can then figure out how many cells there will be at the end. Or if I tell you how many cells there are to start and how many to end, you can figure out how many cell divisions there were. Or if I tell you how many cell divisions there were and how many cells there were at the end, you can tell me how many there were to start with. Let's do an example. So let's suppose we had gone to a picnic and we brought a potato salad and it sat on the picnic table in the sunshine for some period of time, let's say 4 hours. And suppose that when we put that potato salad down, there were 10 cells of a particularly dangerous species. There were 10 cells at that moment. And in the conditions of lying in the sun, in this particular nutrient-rich environment, they have a 20-minute generation time. That means they're going to double every 20 minutes. So our starting number of cells is 10. We know our generation time and we know our incubation period, so we can figure out the number of cell divisions. How do we figure that out? Well, we know that they divide every 20 minutes, and we know that we have got 4 hours. There are 3 divisions per hour times 4 hours gives us 12 cell divisions. So 12 cell divisions is the number of times these 10 cells are going to get a chance to divide. So what is our ending number? We start with our 10 cells. We double them 12 times, 2 to the power of 12, right? 2 times 2 times 2 times 2, 12 times. So 2 to the power of 12 is 4,100-ish, times 10 is about 41,000 cells. And this, going from 10 cells to 41,000 cells, it's massive in amplification. And it is the reason behind the 4-hour rule. If you have worked in food service, you know this. You can't leave food out at like room temperature, for example, and serve it to people after 4 hours because it's not safe. That whatever this is might be a tolerable number of pathogens at 1 o'clock is no longer a tolerable number of pathogens at 4 o'clock or 5 o'clock. All right, so let me just pause here for a moment and check in. Did this make sense? Yes, no, or kind of. Wonderful. Some of you said we need practice. Well, guess what? You're in luck. I came ready to practice with you. We only have time for some of the examples and then we'll pick this up next time. So let's start with this one. At 1 o'clock, a refrigerated chicken breast had 100 salmonella cells. Now, your fridge, unbeknownst to you, breaks, the temperature goes up. And at this new temperature, the generation time is 1 hour. Okay, that means every hour, this population is going to double. Let it go till 5 o'clock. Okay, so it starts at 1, goes to 5, so 4 hours, doubling every hour. How many cells will there be at the end? Put your answer into the chat, put your math in if you can. Okay, I'm going to give you a second to do this. All right, let's see how we did. Beautiful. So many of you used the equation. You started with our n sub 0, our starting number of cells is 100, and our number of cell divisions, and if it's a 4-hour time period and the generation time is 1 hour, there's going to be 4 cell divisions. So 2 to the power of 4 is 16 times 100 is 1600. Now, if this equation is tripping you up, you can do it another way. You can say 4-hour incubation period, generation time is an hour, so that means 4 divisions. And so 100 multiplied, or 100 doubles to 200 in the first division, to 400 in the second, to 800 to 1600. So you can just do it quick like this. What is 2, Esther? Can you say more? 2 is just part of the equation. Let me go back. So in this equation, you can see that 2 is a part of the equation. So whatever our starting number of cells is, we multiply it by 2 to the power of our cell divisions. Okay, so we're doubling it, basically. Are we doubling it once, 2 to the power of 1, or doubling it 10 times, 2 to the power of 10? Okay, we have time for another example. And this one, we're going to actually address how long does doubling take. So figure out our generation time. So let's suppose we had a restaurant burger and there were 5 E. coli cells at 3 o'clock after it was cooked. And then we forget and we set it under the kitchen heat lamp. We forget all about it. And it accidentally gets served as this dried up old husk at 6 o'clock when the dinner rush happens. At which point there are 160 cells. Now, I'm going to tell you something that you don't have to memorize, and it's a concept not relevant to this current information, but we'll come back later and it helps us to understand why we would do this math. In the pathogenesis section, we're going to learn something called ID50, the infectious dose. That's how much of a pathogen do you need to be exposed to have a 50% shot of getting sick. Okay, ID50, infectious dose. So the number of E. coli 0157H7s that are likely to get about half of the people who are exposed to it sick is 100. Okay, this information is how we sort of determine where we set our tolerable limits. Okay, so when you eat food in a restaurant, it's not sterile. There's bugs in it. It's just a small number. I don't mean like insects. I mean like microbes. There's bacteria. There's a small number. And when they sanitize surfaces and so on, there's a number that you have to be below. Okay, it's not that you can get down to 0 necessarily. All right, so if this is 100, then 160 is serious and 5 is probably not serious. So let's calculate the generation time. How do we do this? We need to use our equation. So we know that we start with 5 cells and we end up with 160. How many divisions did it take to get here? Well, we could do it really simply on the back of a napkin. Okay, that's how I'm going to describe it. Or you could use the equation. So in the first division, 5 goes to 10. 10 goes to 20, that's the second. 20 goes to 40, right? That's the third. To 80, to 160. That's 5 cell divisions. And we have 3 hours, okay? And so we take our 3 hours and divide by 5 to get 36 minutes. Now, another way to do that is with this equation, right? We could say our ending number is 160, our starting number is 5, 2 to the power of x. We're trying to solve for x first. 160 divided by 5 is equal to 2 to the power of x. 32 is equal to 2 to the power of x. What does x have to be in order for 2 to the power of that number to be 32? 2 to the power of 5 is 32. So you could do it that way. For this, you would need a calculator that allows you to solve for exponents. Or you can do it this way. You can go 5 to 10 to 20 to 40 to 80 to 160, 1, 2, 3, 4, 5 divisions. Then you go, okay, now I know how many divisions there were. I know my whole incubation period is 180 minutes. Divided by 5 is 36. Did that make sense? Yes, no, or kind of. It's fine if it didn't. You'll have chances to practice this, but just be honest. Great. Okay, so when we come back in our next session, we're going to start here. And we're going to do a few more examples. You're also going to have a chance to do these types of calculations in the practice quiz. There's a practice quiz for the growth practice problems for this section. I highly encourage you to do that, especially if some of this was feeling a little like, and then we've got a little bit of stuff left to talk about before we start metabolism on Wednesday. Let me just scooch forward and yeah, I'm going to stop the recording here, but I do want to talk to you about what to do before we meet again.